Awesome. Well, thank you all for being here. As Susanna said, my name is Zoe Todd. I am an assistant professor in the chemistry and astronomy departments affiliated with WiCOR here. It is my immense pleasure to introduce Jack Szostak to give today's lecture. So Jack is currently a university professor at the University of Chicago, where he moved in 2022, 3 years ago, in the Department of Chemistry. Before that, so Jack got his BS from McGill University in cell biology in 1972, where he started college at the bright young age of 15 years old. And then he received his PhD from Cornell University in biochemistry in 1977, where he worked in Ray Wu's lab. Then he stuck around Cornell for a couple more years after that before moving to Harvard Medical School as an assistant professor there. And I tried to look up all of the different affiliations that Jack has had at Harvard over the years. So it's like Department of Genetics and Department of Chemistry and Department of Molecular Biology, many of those, but he's at Chicago now in the Department of Chemistry. Jack has many numerous awards and honors, including, last but not least, the Nobel Prize in 2009 in Physiology and Medicine, where Jack was awarded this prize in collaboration with Elizabeth Blackburn and Carol Greider for their work on telomerase and telomeres, so trying to understand why chromosomes don't degrade every single time that they replicate overall. Jack is also a fellow of the AAAS, of the Royal Society, the American Physical Society, and the National Academy of Science, and I'm sure I've left off many other accolades from Jack as well. Scientifically, so I already mentioned Jack's work on telomeres and telomerase that earned him the Nobel Prize in Physiology and Medicine, but Jack has also been involved in things like DNA recombination and the double-strand break model in meiotic division, another huge discovery. Jack also was involved in developing in vitro evolution, a technique that is now used widely across biochemistry. And last but not least, also developing aptamers, which are different RNA and peptide binding ligands and sequences there. So a very historical and storied career from Jack Szostak overall. And since about 2000, Jack has pivoted his lab's work to try to understand origins of life on Earth, and that's what we're going to be hearing about today. So hopefully I did not get too much of that wrong. How'd I do? Okay, awesome. So I will turn it over to Jack. Please take it away, and thank you very much. And let's welcome Jack.
Okay, thank you very much, Zoe. What do we? Yeah, everyone can hear me? Okay, all right, good. Yeah, so thanks, Zoe, that was great. And I also want to thank both Zoe and Susanna for inviting me to give this lecture, and then I'm giving a more technical talk tomorrow about more of the RNA chemistry side of things. I did want to mention before going on that, so I was in the Department of Molecular Biology at MGH, and I never ever thought I would have an astronomy student doing experiments in my lab, but Zoe did a good part of her thesis doing experiments in my lab as a student of Dimitar Sasolov. Okay, so I want to give a kind of overview of our sort of current picture of the origin of life, and so it'll be at a kind of very broad overview. What we want to understand is the sort of the whole pathway, from planet formation, which I won't say much about, but chemistry on the early planet, how that gave rise to the building blocks of biology, and eventually simple cells, maybe something like what you see in the schematic version on the slide, and then onto something that can start evolving and eventually give rise to us. So before I go into that, so work on how life got started is really closely tied in with the question of whether or not there's life out there, exoplanets. And I like to begin talks like this by asking you, how many of you think that life is common in the universe, that there are like lots and lots of planets out there that have life? Okay, so yeah, so lots of people. how many of you think that life is like so complicated and so hard to get to that life could be very rare? And see, there's also lots of people who take that point of view. And I find it very interesting that, we don't know the answer, right? That's why we're all trying to do all of this work and understand how life got started. And, if it looks like a really easy pathway, then life must be common. And if it looks like there are bottlenecks or lots of hard steps, maybe life is really rare. And so people like me and my colleagues, we're waiting for our astronomer friends to give us evidence of a second independent origin of life, which will really prove that it's not that hard. And conversely, we hope to, experimentally see if it looks like there's an easy path or not, which might inform astronomical efforts. Okay. So there are obviously very good reasons for thinking that, life might be abundant in the universe. I mean, there are so many places, right? I mean, there must be millions of Earth-like planets orbiting sun-like stars in our galaxy alone, and there are, many trillions of galaxies out there. So lots of possible places. We know that most stars have planets now. So there's got to be a lot of places that could support life. And what we don't know is whether they actually do support life. Okay? And the other reason for thinking that life might be kind of as, I think it was Christian de Duve said, a cosmic imperative, is there was also lots of time, right? Even on our planet, there's a period of 500 to 700 million years between when the Earth cooled down enough to have liquid water on the surface to the earliest really clear signs of life on the planet. So hundreds of millions of years. So a lot of places, lots of time, maybe life is common. On the other hand, if you look at modern biology, it's crazy, right? It's so complicated. There are thousands of chemical reactions all going along in parallel, not interfering with each other, catalyzed by thousands of complex enzymes. And it's hard to imagine how something that complicated evolved spontaneously. But to me, actually, the thing that is the most amazing is if you look at the organization of life today, it's extremely self-referential, right? So we have the central dogma pathway here, but the point is that every component of this system depends on every other component, right? So if you want to transcribe DNA into RNA and translate it into proteins, so you need enzymes to do the transcription. You need complex machinery to make proteins. You need the metabolites that are made by enzymes. And similarly, to make proteins, you need DNA and RNA and metabolites. It's hard. It was for many, many years, decades. It was hard to imagine how such an interdependent system could arise spontaneously. Okay. So of course, there were a few really smart people in the 60s who, following the initial discovery of the three-dimensional folded structure of tRNA, kind of realized that the molecule in the middle, RNA, could potentially act as an enzyme. So people like Leslie Orgel and Francis Crick and Carl Woese posited that RNA might be able to act as an enzyme. And nobody took that seriously for about 15 years until Tom Chuck and Sid Altman discovered actual ribozymes, RNA molecules that are enzymes in modern biology. And that, of course, gave rise to a much simpler view of what early life might have looked like. Okay? Basically, so we call it the RNA world in Wally Gilbert's little phrase. But the idea is you would have cells that are RNA-based in terms of their biochemistry, right? So they have an RNA genome that is replicated by RNA catalysts, could even make metabolic ribozymes with regulation by riboswitches. And so in this sort of simple view, you don't need coded proteins or peptides at this very early stage in the evolution of life. Okay, so now, so now we have. So let me just say one more thing. So the sort of smoking gun for this view of early cellular life is the structure of the ribosome. Okay? So this is a view of a crystal structure of the large subunit of the ribosome from work by Tom Steitz. And what you can see is all the gray squiggles are RNA, the gold squiggles are proteins, but where the chemistry happens in making peptides is where this green transition state analog is. And so there's no protein near it. So all of the proteins in every cell in our bodies, and every cell of every type on the whole planet, every protein is made by RNA. Okay, so I think the only logical inference from that is that RNA was here first, and through a process of evolution, gradually developed the machinery to do coded peptide synthesis. Okay, and there's a lot of interesting problems in trying to understand what the sequence of events was there, but it's been very hard to figure out. But there are a lot of interesting challenges. Okay, so now we have a simpler view of what early life could have looked like. And so now you can sort of restate or rethink the problem of how life got started in a simpler and more direct way. So again, we try to break these big problems down into somewhat simpler chunks. So one of the key questions is, how did the chemical building blocks of biology arise? So we need nucleotides to make RNA, and nucleotides are pretty complicated molecules. So there's a lot of challenges in trying to understand how molecules as complicated as nucleotides could actually be formed spontaneously on the early Earth. Once you have all the right molecules, then you have a different set of challenges, right? How do a bunch of molecules get together and start acting like a cell that can grow and divide and replicate its genome and start evolving in interesting ways? And then something that has become increasingly an important part of the field is trying to understand the actual environments on the early Earth where these things could take place. So understanding the geochemistry and the geophysics is obviously a key part of the overall question. Okay, so back in the 1950s, when Stanley Miller was a young graduate student in Harold Urey's lab, he did the now famous iconic experiment of passing a spark discharge through a simulated early Earth atmosphere, which was a very reducing atmosphere, and found that they could make a whole bunch of molecules, including amino acids. Right? So at the time, it was a huge breakthrough, because just the idea that these sort of iconic building blocks of biology could be made in such a simple way was a huge breakthrough, and in many ways is the starting point of modern prebiotic chemistry. Okay. And you see actually similar things. Let's see if we can start this. So you may have heard of the sample return mission from the asteroid Bennu. So we were able to bring back samples from this asteroid, and people have been busily analyzing the organic chemistry. And again, you see amino acids, you see the nucleobases, the part that are the information-bearing parts of nucleotides. And so from these two things, a lot of people draw the conclusion that this kind of simple chemistry provides an answer to how you make the molecules of life. And unfortunately, I think that's not the right way of looking at it, because in both the Miller-Urey experiment and in meteoritic and asteroid chemistry, what you find is a complex zoo of compounds, thousands, depending on how sensitive your analytical techniques are, right? You see more and more different compounds. And the ones that you would want are usually parts per million or parts per billion, and it goes on down from there. So there's a huge variety of compounds, most of which are not part of biology and are not useful in building biology. And there's trace quantities of molecules that look relevant to biology. And one of the most interesting things that came out of the analysis of the Bennu amino acids is that they're racemic. In other words, they have equal amounts of the right and left-handed mirror image forms of those molecules, right? And the people who found that result were actually quite sad, because they expected, on the basis of earlier work that had been done on meteorites from Earth, they expected to see an excess of the isomer that you find in biology, but it's not there. So the lesson that I draw from that is that the chemistry that gave rise to the molecules of life happened on the Earth, okay? And so this is all very intrinsically interesting chemistry. A lot of it is probably really important in terms of understanding planet formation. But I think that most of the relevant chemistry happened on Earth. And what we need to understand how that chemistry could give rise to biology is to have pathways that give you a large amount of a small number of compounds. We want the right compounds and not millions of irrelevant compounds. So that's really the challenge in understanding chemistry that could lead to life. Okay, so my favorite compound in that regard is cyanide. And I like that because of the irony that something that's so poisonous is probably actually the fountain of life. Okay, so there are many ways to make cyanide. And cyanide is found in protoplanetary disks, molecular clouds. It can be generated in the atmosphere of a planet in many different ways, both through impacts, UV chemistry, lightning, and it's a high energy compound that you can use to actually to make many of the molecules of life. And so I want to show you a few pieces of how that puzzle's been unraveled in recent years, not by me, but primarily from the lab of John Sutherland in the UK and some of his students and colleagues. So the problem with cyanide is if you make cyanide in the atmosphere, it can rain out to the surface, but it will rain out as a very dilute solution in water, right? And dilute cyanide is pretty useless because all it's going to do is sit around and hydrolyze and eventually give you ammonium formate, which doesn't go anywhere. All right. So question is, we have this molecule. We know that it can be very useful, but how do we use it? How do we build up a reservoir? And so what looks like a nice model is if you think of a region on the surface of the Earth that is has a lot of geothermal activity, so say a volcanic area, something like Yellowstone or places in Iceland, or an impact crater where there's a lot of shattered rock and you have water circulating through the shattered rock. That water circulation will bring up ions that have been leached out of the rock. And among these ions will be ferrous iron. And the interesting thing about ferrous iron is that it will complex very rapidly and tightly with cyanide to make ferrocyanide. Okay? And so this compound, so various salts of ferrocyanide, can build up and essentially precipitate along with sediments, either if the lake that this is happening in or pond dries out, or just by building up. So David Catling's done beautiful studies of alkaline carbonate lakes, which provide an environment where ferrocyanide salts can build up to high levels and can accumulate in sediments. Okay, so that's what's supposed to be represented down here. So you're building up a reservoir of cyanide in the form of these ferrocyanide salts. Okay, now imagine this pond or lake dries up, and there's a lava flow over it, or maybe an impact that heats things up, or heat from underlying magma. That can start to bake that cyanide reservoir, which could be meters thick of cyanide. And it turns out if you heat these ferrocyanide salts to different fairly high temperatures, temperatures you can get to from, say, lava overlying or underlying lava flows, you can release the cyanide from its iron complex. And you can also release other useful things like ammonia and cyanamide and possibly other things. And so now you have this these useful products that have been released from the iron. And now, of course, groundwater will start to seep through this reservoir. And you can imagine this slowly leaching out these compounds from this reservoir. So you can have incredibly concentrated mix of cyanide, cyanamide, ammonia, other things. So it really solves the problem of how do you go from raining out a really dilute solution to now having a really concentrated, possibly molar concentrations of these reactive starting materials. Okay, so if that material comes to the surface in a spring and starts to flow down, so this is a photo from Yellowstone, one of those beautiful terraces. So probably not very prebiotic, but it's a pretty picture. You can imagine a shallow stream. Now it's going to be irradiated with ultraviolet light from the young sun, and we know that UV light will come down to the surface because there's no oxygen in the early atmosphere, there's no protective ozone layer, so UV can come down, and UV light will start to photochemically process this really concentrated stew of starting materials. Okay, and so we know very well the chemical pathways that will process these starting materials into simple sugars. And then these simple sugars can react with, in this case, cyanamide to make intermediates like this. And the next step gives you this more complicated intermediate. And the beautiful thing is both of these intermediates can actually be purified very easily. So that first small intermediate, it turns out it sublimes with just gentle warming. So if there's a cold surface nearby, you could get pure crystals of that compound. If it then goes on to react to sort of, say, be redissolved and react with the next sugar, then you make this, the next intermediate, which actually makes these incredible, beautiful crystals. And the nice thing here is you're crystallizing out the molecule that you actually want to go on to make nucleotides. And all the other stuff, in principle, could get washed away, just leaving you with a purified reservoir of the starting material for making nucleotides. Okay? And actually, the story is even better than that, because in recent work from Furkan Öztürk and Dimitar Sasalov, that same compound has a somewhat unusual but very important property in that when it crystallizes, the right and left-handed versions of the molecule make separate crystals. And if that crystallization happens on a magnetized surface in a magnetic field, you get selective nucleation of crystals of one of the enantiomers. So it's actually a potential way to get not just the compound you want, but the right mirror image form to go on to make nucleotides that are now not a mix of right and left-handed forms, but just the one that we see in biology. Okay, so I mentioned that UV light plays important roles in the chemistry. And this is a really beautiful and surprising separate role for UV light. So in the pathway, so starting from that nice crystalline compound, so 2 steps later, you make something that looks like this. It's like a nucleotide, but this blue part is below the sugar part, which is shown in green. In biology, we want the blue part, the nucleobase, to be above the sugar ring, right? And it turns out that flipping from below to above is driven by ultraviolet light. Okay? So you have all these photochemical steps that are essential for making the molecules that we need to build biology. So that is one of many, many reasons that life had to emerge on the surface of our planet, right? And if you see any of this stuff about life starting in deep sea hydrothermal vents, it's just completely wrong. Okay? In fact, to me, it's a great example of pathological science, because people have been talking about that model for 40 or 50 years, and there is no evidence, and nobody has ever been able to make any interesting compounds that are on the way to biology in that environment. And yeah, it's very frustrating that something that is so obviously wrong continues to get propagated in the public press. Okay, so from people who are serious scientists, we have many lines of evidence that life started on the surface. Okay, now one of the long-standing puzzles in origin of life chemistry had to do with phosphate, right? We know that phosphate had to be around. Phosphate is an integral part of the backbone of RNA and DNA. So you can't start life without phosphate. But everyone used to say, well, this is a problem because phosphate gets precipitated as an insoluble mineral, apatite, in the presence of calcium. So for decades, people are saying, how are we going to get phosphate? Where's the phosphate going to come from, right? How do we get it out of apatite? And it wasn't until David Catling came along and actually started thinking about it seriously and creatively, and actually looking at places on the modern Earth. There's this lake in British Columbia that has up to 40 millimolar of free phosphate. And why is that? It's because of several reasons, but one is that the phosphate is, it's essentially, there's very little living stuff there, so the phosphate doesn't get soaked up and removed by biology. And also the chemistry is such that the calcium is precipitated, and that allows phosphate to accumulate. So if you're in an environment like an alkaline carbonate lake, you precipitate the calcium, phosphate can be leached out of rocks, it can accumulate to high levels. We can have the phosphate at concentrations that we need to do the prebiotic chemistry to build up to things like nucleotides and RNA. Okay, so that's all I'm going to say about making the building blocks. Now I want to talk just a little bit about RNA chemistry, and I'm going to do that a little bit quickly, because the more heavy-duty organic chemistry will be in my talk tomorrow. So if you want to hear that, you can hear about it tomorrow. So this will be a more high-level view. But one of the, I think, early breakthroughs that always impressed me came from the late Leslie Orgel, who was one of the real pioneers in this field of prebiotic chemistry. And Leslie made many contributions, but I think one of his great insights is that the molecules that are found in biology that are used as the substrates to make RNA and DNA are these types of molecules. They're nucleoside triphosphates, right? So they have this chain of phosphates, And if you have molecules like this, they're great if you have some sophisticated catalysts. Like you have enzymes that can accelerate reactions by, a billion or a trillion fold, right? But if you don't have a really powerful catalyst like this, then these types of substrates are pretty useless because they'll just sit around and they won't react and they won't do anything. So what you need to get life started is something more like this type of molecule. So now we have this imidazole derivative here, and these molecules are much more reactive, okay? So it's both good and bad. And the good side is they will actually start to polymerize all by themselves, either without a template or, more interestingly, with a template. You can start to copy the sequence in a template strand with molecules like that. The bad side is they tend to react with water more quickly, so they hydrolyze. So it means now we have a new chemistry problem, right? We have to understand how to make these and sort of regenerate them so that everything doesn't just run down by hydrolysis. Okay. So and that's still a very active area of research. Okay. So I just want to show this video that was made by Jano Dewasa many years ago, and it shows how we used to think about copying RNA strands. And the idea was you'd have a strand of RNA floating around in a sort of rich chemical environment. And these activated building blocks would find their partners by base pairing and sort of click together to build up a complementary strand. Okay? So we really thought for a long time that that's how it ought to work, because that's more or less how it works in biology, right? You have activated nucleoside triphosphates, they come in, an enzyme adds them one at a time to a growing chain. So it turns out that actually doesn't work very well. And the way the actual chemistry of template copying is something that's been worked out over the last 10 years or so, and it works in a surprising and different way. But I'm not going to go into that too much today. I wanted to instead just focus on a different problem that was pointed out by Jerry Joyce and Leslie Orgel more than 30 years ago. And that was a lot of us molecular biologists were doing these experiments trying to look at how RNA could get copied without enzymes, right? But we're doing it in a totally artificial way, using like high concentrations of purified molecules. And they basically they made fun of it as the molecular biologist dream, that there would be some like like Darwin's warm little pond, except full of concentrated, pure, activated ribonucleotides, right? Which, if you think about it, is somewhat ludicrous. And so the question is, if you're starting off with a mixture of things, how do you get to RNA that looks like the RNA you find in biology? So that's something we started to work on in my group several years ago. And I'll just show an outline of the type of approach we used, which was, okay, so here's the puzzle, right? You might have in your, in your test tube or in your warm little pond, a mixture of different types of nucleotides. So ribonucleotides are the building blocks that will give rise to RNA. But when you're making ribonucleotides, it's actually hard to avoid making some of the related molecules like arabinonucleotides or 3O-nucleotides, even deoxynucleotides that are the building blocks of DNA. So you have all this stuff around and it all gets polymerized together. You're going to make short chains that are really heterogeneous, right? And that doesn't seem like a great place to start, right? Because something like that, even if you could replicate it, you would always have this heterogeneous mixture, and yeah, things wouldn't fold up properly. It just seems like a bad way start trying to get to an evolutionary system. So we started to look at this kind of systematically. And the idea was that if you have these kind of random sequence chains, just by chance, pairs of them can come together in sort of overlapping configurations. It would be set up to do a little bit of chain elongation, primer extension, right? And so then we can look at, well, how well does that work with the RNA building blocks, or the arabinonucleotides, or the threonucleotides? And everything is worse than RNA, right? So the building blocks of RNA always win out. They're better at doing copying chemistry. If you put some of these other things in, like arabinonucleotides, you can add one to a growing chain, but that kills it. It's totally dead. On the other hand, you can copy over these heterogeneous chains. And what we'll preferentially do when you copy over it is to incorporate ribonucleotides and make more RNA. So if you keep doing that, if you have cycles of association and dissociation, little bits of primer extension, it's RNA that's modern-looking RNA that's going to accumulate. So we think this is a kind of simple process by which you could start with a bit of a messy mixture, and by doing this kind of cycling, end up with something that is increasingly homogeneous and looks pretty much like modern RNA. Okay? So, which we think is the ideal starting point for an evolving system. Okay, so that sort of feeds into our overall view of what primitive cells would have looked like. So a simple cell membrane and encapsulating short chains of RNA that can get replicated just using this simple chemistry, no enzymes, nothing fancy. And so a lot of what we're working on is how to go beyond just copying a template strand to actually doing cycles of replication, right? Like what you see in biology, but in a much smaller scale and without any evolved machinery. Okay. Now, the other part of it that I haven't talked about at all so far is the membrane part. What are these primitive cell membranes made of? And for modern cells to grow and divide, it takes a huge amount of evolved machinery. Right? But obviously, at the origin of life, there was no evolved machinery. So how can you have a simple physical system where things are growing and dividing and replications happening on the inside? And so I want to talk about that for a little bit. And I just want to begin by pointing out that it's actually a really remarkable thing that very simple molecules, like simple fatty acids, like shown here, if you just shake them up in water, not too acidic, not too basic, a little bit of salt maybe, they'll spontaneously assemble into structures that look kind of like cells. They're membranes, they're bilayer membranes. Here you have this really beautiful image of there's a big vesicle with smaller vesicles trapped on the inside. And it's just a spontaneous self-assembly process. These spontaneously assembled vesicles have really interesting and amazing properties. Because these are single-chain lipids, not unlike the molecules that make the membranes of our cells, which are have two chains. They make much more stable membranes. These membranes are very dynamic. The molecules that make a membrane are leaving and coming back in like every second or so. It's a very dynamic system. And yet the vesicles themselves maintain their individual identity for weeks or months. So what we did in this experiment was make two different populations of these simple fatty acid-based vesicles. We labeled them with different dyes. Then we mixed them together and then photographed them in the microscope. I think this was after a day. And they're not mixing. right? You still see red and green vesicles. There's no exchange, even though the molecules that make up those vesicles have exchanged many times over that time period. So they maintain their individual identity, which is a critical thing for Darwinian evolution. Right? If everything is just in solution or everything is fusing and mixing, there's no way to have an evolutionary process. So this shows that these really simple structures can do that. And then the next thing we want to know, well, okay, so somehow they have to be able to grow and divide. So how can that happen? Right? So we studied that for years in very, very indirect methods. But we always wanted to see, like, how does something like this grow? We had to watch it in the microscope. And it wasn't until a new graduate student joined the lab with a very different background from everyone else. He was actually trained as a mechanical engineer. And he came into the lab and like quickly figured out how to do it. And the result just is completely mind-blowing. It's still mind-boggling, even when I look at it today, 15 years later. So what you're seeing here is one of these vesicles. We're looking at it in the microscope. It's got a fluorescent dye on the inside, so we can see what happens. Then we're adding more food, basically more fatty acids to the system, and we wanted to watch this thing grow, right? So we thought, oh, it's just going to slowly get bigger. Maybe it'll change shape a little bit. We didn't expect what happened. Now I got to try to lower the lights. Let me see. System off. That doesn't sound good. No. There was a way to do this on this little. Okay, that's perfect. That's perfect. Okay. All right. So we're going to add food and then watch what happens. And what you can see is this like faint filament growing out. And, oh, that's, okay, I haven't seen that straight background. Anyway, you can see the spherical vesicle transforming into this long, branched filamentous structure, right? It was not what we expected. I almost fell off my chair when I saw this. And so we have some ideas about how this happens, but it's still, they're mysterious. This is just soap and water, right? And yet there are complicated things like this that happen that we still don't fully understand. And now let me show you another thing. So now, one nice thing about growing in this way is these filaments are very fragile, right? It doesn't take very much to lead to division. And so I show you another video of that. So first of all, you have this weird thing where they form sort of a string of pearls type of shape, and then a little bit of disturbance on the surface. You can imagine wind blowing over a pond is enough to snap the filament, and now you have daughter protocells, daughter vesicles. So eventually, over time, these will round up, and you would have 2 or sometimes 3 or 4 daughter cells from one parental cell. And what makes it even more interesting is there's different ways this can happen, right? So here's another way that you can get division. And this happens from a photochemical process. It's driven by UV light. So you get this really extreme, what we call purling instability. So it's like a string of beads. And eventually these beads separate. So this way you can get dozens or 100 daughter protocells from one initial protocell. Okay, so in fact, there's even more ways that you can get growth and division. So I'm going to show you in a minute a video that was taken by Anna Wang when she was a postdoc in my lab. And so this is a different model system where here these are larger spherical vesicles. They have a single membrane layer. And again, we can feed them with more fatty acids. And what you'll see is you start to see shape changes. So the surface is growing. That allows the shape to start doing all kinds of weird fluctuations. And you can see spontaneous budding and division. So once again, just by the act of feeding these vesicles, you can have growth and division into daughter protocells. So if you think about a primitive cell with one of these very simple fatty acid-based cell membranes, there are multiple ways that growth and division can happen. We've been able to show that we can do some RNA copying chemistry on the inside of these vesicles. And what we're focusing on now is trying to get the whole thing going, getting cycles of RNA replication going on inside vesicles that can also be growing and dividing. And I think at that point, we'll have a really nice model of what early cells might have looked like. And the hope is to watch the system start to evolve and watch new RNA sequences arise that can do something useful for the cell that they're in. Okay? So I mentioned at the beginning that we've got all of this prebiotic chemistry. We can make the building blocks of biology. We can assemble them into RNA chains, into membranes. Where on the primitive Earth would this kind of thing be happening? Okay? And so the kinds of environments that we like are either volcanic areas. So this is a photo of Yellowstone Lake. We like this because areas where you have ponds or lakes, they allow things to accumulate, right? So materials can accumulate over time. They can get, in volcanic regions, they can get concentrated by wet-dry cycles, and you can have fluctuations like heating and cooling, or freeze-thaw cycles. This is another great way to sort of kickstart a lot of interesting chemistry. But since you're on the surface, you have access to energy from ultraviolet light. And one of the key puzzles that still remains, that's being worked out, but it's still not well understood, is how do you harness the energy of ultraviolet light to make in a useful chemical form? So you can have a way of transforming that energy into chemical energy that can drive reactions. Okay, so that's a really important current area of work. But these kinds of surface environments are I think, very promising. And you have very similar environments around impact craters, right? So again, you have heat from the impact, you have fractured rock, you can accumulate things, a lot of the same kind of geophysical properties. So again, a few images from Yellowstone. So in some of these hot spring pools, you can actually see things crystallizing around the rim. So you probably remember seeing these things. I mean, it's so good to get out of the lab and out into nature and actually get some idea of what, even though the modern Earth is so different, right? We can get an idea from looking at environments like this for situations that could have arisen on the early Earth. And again, here's one of the smaller geysers. So you'll have periodic outbursts of hot water, and so you can have rapid temperature cycling and things like that. And so that gave rise to one kind of model for how the environment can drive the primitive cell cycle, right? So this is important because in modern biology, cells have such complicated, evolved machinery. They have a lot of control over when they grow and when they divide, right? But a primitive cell doesn't have any of that. It's totally reliant on fluctuations in the surrounding environment to allow it to grow and divide or to drive its cell cycle. So this is just one idea that we came up with, I think this is almost 15 years ago. But in an environment like Yellowstone, you have these hydrothermal vents that release streams of hot water that rise and then get dispersed into the surrounding cold water of the lake. So if you had protocells living in such an environment, right, every now and then they could get swept up by one of these streams of hot water. That could drive strand separation. The strands of RNA could come apart. The membranes become more permeable, nutrients can rush in, but you don't want to cook RNA, it's very delicate, so they very quickly get swept back out into the surrounding cold water. Now the membranes become more sealed, strands come back together, you can do some copying chemistry. So we think this kind of environmentally driven cell cycle, something like that has to be what went on at the very beginning. And then gradually over time, cells evolved the machinery to give them more control over survival in different environments, and also sensing the environment to know when is the right time to grow, when is the right time to stop, when's the right time to divide. Okay. So then just to sort of wrap up, we can say, so from all of this work, right, many labs over many years, what have we really learned? And to me, so we've learned lots of facts, right? And there are lots of like interesting small advances in the chemistry. But to me, the bigger lesson is that in many cases I've wondered, like, why didn't we figure this out 30 years ago or 50 years ago, right? Why has it taken so long to work out a lot of this pathway? And I think it's because we're often held back from making progress because we have preconceptions and biases about how things ought to work, right? You see how things work in biology and you think, well, maybe it should work that way at the beginning, but it doesn't have to, right? Or maybe you have a model that you've developed yourself for how things ought to work, and it's very hard to let go of your own model, right? So I think to really make progress, you have to let go of preconceptions, and I think doing that in this field has led people to discover all kinds of crazy, unanticipated physical and chemical phenomena. So I think this field is great because it's a goldmine for discovery of surprising new phenomena. And I think by continuing this kind of work, I think over the next decade, we'll actually get to a point where we have a pretty good understanding of at least one and possibly many ways that life could have started when our planet was young. Okay, so I have tried to mention a few of the people involved as I went along, but of course, all this work was done by fantastic students and postdocs, and you can see from the faces and the names that these are people from all over the world. Marco's from Italy, Aman's from India, Daniel's originally from Hungary, China, England, China. So it's a very international effort, and we're always looking for people who are, who have the determination and the energy and the initiative to do creative new things and work on hard problems. And it's been really great working with people like this over the years. And so if you want to learn more, so we've raised a lot of interesting questions, right? There are many unsolved mysteries in this field. And I just want to make the point that if you want to get the answers to these questions about how life got started and other questions in science, you have to stand up for science. You have to make your voices heard. You have to fight back against all the crazy stuff that's happening these days in terms of defunding science and abandoning the rule of law. And we're not going to have a better understanding of how things work unless people fight back against that. And I'll stop there. Thanks.
All right, thank you so much, Jack. That was fantastic. We have time for some questions. So if you have a question, if you just want to raise your hand, and either Vanessa or I or Susanna will run around. So, okay, let's tag team them.
Let me also just say that I did write a book recently, so another way to learn more about this is you can read that.
Everyone go buy the book.
Thank you so much for the great talk. The question that I had was your concluding point on fluctuations.
Yes.
So I was wondering if are there certain timescales that that fluctuate, like, are there certain more conducive timescales that fluctuations can be conducive, or, I mean, yeah.
No, that's a great question.
Yeah.
So, so there are things that constrain the timescales, right? And I think that the relevant timescales depend on where you are in the whole pathway, right? So in terms of getting the right geological setting, right, there the timescales can be anywhere from tens of millions of years to millions or hundreds of thousands of years, right? The planet had to cool down. You had to get liquid water. You needed to dry land rising above the ocean. So that's probably a long time scale. But by the time you get to this kind of prebiotic chemistry, making nucleotides, these reactions go in, hours to days to weeks. And so the time scale is speeding up. By the time you get to making activated nucleotides or RNA chains, right, they tend to fall apart. And so the whole process has to happen on a much faster time scale, of maybe hours to days. And we like to think of possibly temperature fluctuations happening on time scales of hours, maybe. So we could do multiple cycles in a day to get one cell cycle. So yeah, it can't be too long. You can't be hot for so long, or you'll destroy the RNA. You can't, if you're cold for too long, maybe that's okay because nothing much will happen. But yeah, we want fairly fast fluctuations.
Great. Next question over here.
Does the RNA replication need to happen in water, or could it happen in the atmosphere of a gas planet?
So everything that we've looked at is in water. And yeah, I can't really see how it could happen in an atmosphere. Maybe if there were water droplets in an atmosphere, something like that. But yeah, all this chemistry pretty much has to happen in water. It's a great question. Thanks.
Great talk. I have so many questions, so I'll try to keep my favorite one. So all life on our planet kind of seems to evolve from the same place. Do you think the event that created that has happened once, or do you think it's happened multiple times and just some have failed?
Yeah. So all life on the planet, right, traces back to a common ancestor. But that common ancestor was already pretty much like modern bacterial life, right? It is very distant from the origin of life. So for all we know, we can't tell whether the actual first cells that emerged, was that an event that just happened once, or if it could be that life was popping up all over the early planet. Maybe most times it got destroyed by some environmental fluctuation and impact or something. We can't tell because that part of the history is lost. All we know is that there was a surviving lineage that gave rise to a common ancestor at a much later time. All the other branches of the tree are gone.
Thank you.
Thank you, Professor Szostak. That was amazing. I'm from the geoscience department, and now I know I'm going to have to be careful not to make the claim that the hydrothermal model is the most accepted one for the origin of life.
Maybe the most accepted, but that doesn't make it right.
Yeah, or it's not the right one. But another thing related to geoscience is I get the feeling that ongoing tectonic activity of some kind sort of rises up as a key ingredient. I mean, impacts, right, could obviously produce transient heat, potentially long-lived. But, for exoplanets, I mean, are we thinking of exoplanets that have tectonic activity as sort of a requirement or an assumption to at least a working assumption?
Yeah, so I mean, yeah, these are all like really interesting questions that I think are quite open. Do we actually need continental crust to provide the right environments, or could you do everything on volcanic island arc type environments? Yeah, we don't know. I mean, these are all, frontier questions for right now.
Questions?
Hello. Thank you very much for this great talk.
Thanks.
So I noticed that you mentioned somehow you're trying to find pathways that can make a lot of relatively pure molecules of specific types so that they are similar to what life is using today. But actually, in my research, I gradually felt that if I want to make these kind of complex molecules, it was more likely that I will make other types of simpler molecules that are more abundant on a prebiotic Earth. And then actually some of them might function similarly as life as today. So it's like an alternative forms of life. So what do you think of the idea that there are alternative forms of life that behave differently and use different things as it today, and then gradually evolve to what it looks like today?
Yeah. So that is a really interesting model that I think there's an interesting bit of the history here. So for a long time, when people, like RNA is so complicated and people couldn't understand any way that you could potentially get to RNA, it really drove a search for alternatives that might be easier to make, easier to get to. And as far as I know, so far, nothing like that has been found. If there is a way to make something that's simpler and easier as a way to get started that could then somehow ultimately give rise to RNA, that would be great, right? So it would be really fantastic to find something like that. We thought for a while that there was something. So there's a nucleic acid called TNA that has a 4-carbon sugar, and it looks and could be easier to make than RNA. And we've looked really hard to see if we can get TNA replication or even copying, and it just doesn't work. So it's kind of interesting, and maybe it's surprising that you have things that do look simpler, but we haven't been able to show good copying chemistry. And so so far, RNA is the only thing that we know that works, but that certainly says we should keep looking for other things. Are there other ways to get life started? Are there simpler ways? Yeah, these are all things that really ought to be pursued. So it'd be very interesting.
Thank you so much for the talk, and definitely thank you for taking questions. I was wondering. I'm back here.
Oh, okay.
You're all good. I thought it was really interesting when you brought up that the pond or the puddle that life might have started in had a lot of different nucleotides that were not necessarily the one that we see ubiquitous in life today.
Right.
It made me wonder, when you're trying to model experiments, modeling the environment of prebiotic Earth, do you worry about like dirt or like dust or other small molecules that must have been in this puddle where life started?
Yeah. So the way that we approach these questions is we start off with an artificially clean, simplified system, and then we try to work our way back step by step to something that's more prebiotically realistic for things that could happen on the early Earth. So that's why we started trying to look at what would happen if you had all of these alternative nucleotides present. Okay, and so that, I think, we can say that that's not necessarily a problem. Now, we haven't yet gone back to even more messy, complex systems. People like Zoe are doing great work to see, like, would some of those compounds be destroyed by ultraviolet light? What are the chemical and physical processes that might get rid of some harmful interfering molecules? What's the kind of complexity that we really have to deal with realistically? So yeah, I hope that eventually either we or other people in the field will get to that level, but we're not there yet.
Hello, here in the back.
Hi.
Thought-provoking talk, thank you.
Thanks.
You showed the selective crystallization of an enantioenriched material under a magnetic field.
Yeah.
Is there some geological regions where you would anticipate to observe that? And
Yes.
have we found enantiomer-rich material that we believe to not be of biological origin here on Earth?
So none of this chemistry can happen on the modern Earth, right? The environment is totally different, and you wouldn't make molecules like this, and if you did, they'd get eaten up. But the environment that is being considered, and this is from Szostak and Sasolov, and it partly comes from studies of early Mars environments. So if you have a shallow lake that is sort of redox stratified, so you have ultraviolet light. If you have things like ferrocyanide, you can have photooxidation. You convert ferrous iron to ferric iron. You can start precipitating iron oxyhydroxide complexes. You can make magnetite that will, so small crystals of magnetite can form. This has been experimentally shown among other places, Nick Tosca's lab in Cambridge. So those magnetite crystals can sediment out, probably along with other stuff. And from Mars environments, you can actually see sort of flat surfaces that are highly enriched in magnetite. So that's the kind of surface environment that is being looked at to see if we can get this same enantioselective crystallization to happen.
All right, we've got time for 2 quick questions. So one up the front and one more.
Hey, great talk. One thing I was wondering about, and you talked a little bit briefly about the left and right-handed molecules.
Yeah.
I know some people have been looking into trying to create this type of mirror life, right? Like a completely different hand. I know there's dangers to it, and is there a way to do it safely? And and should we even try to pursue it in the first place?
My opinion is that it should not be done. I think if we made mirror versions of a bacterium that could survive in the wild, that could have catastrophic consequences. So I'm one of the authors of that science paper that recommended that this just is something where we just shouldn't go there, right? It's just too risky. And the benefits are negligible, so why do it?
Hi, thank you so much for your talk. My final question is, after listening to everything that you said, I'm very curious what you think about the probability of life yourself. You asked us, and I would like to ask you back. Do you think life is common or uncommon?
So my basic answer is I don't know. We still need to find out more. So it's kind of a cop-out, I know. I can say 2 opposite things. One is there are a lot of chemical steps that we used to think, like, we had no idea how could this chemical step actually happen. And now it turns out it's trivial, right? So a lot of the steps that seemed hard now seem easy. So that sort of goes along with the idea that, well, maybe it's not that hard to get to life, and life might be common. On the other hand, it's the geochemical, geophysical series of different environments. That's what makes me think that life actually could still be rare, right? You have to build up that cyanide reservoir, right? Then it has to be processed. It has to last long enough, and it has to be thermally processed in the right way. And then the materials that come out of that have to go into the right environment for the next steps to happen. And then you have to build up a new reservoir of some intermediate. That reservoir has to build up, has to survive, not be eroded away or washed away or blown away in an impact. And then the environment has to change so that you go on to the next step. And if you have 5 or 6 steps like that all have to happen with just the right environments in the right order, maybe that's just incredibly rare. So I think the geochemistry may be the hard part, not the actual organic chemistry. So yeah, we don't know. I don't know even how we'll evaluate that. Yeah.
What a fantastic question to end our group questions here. If you have more questions, feel free to come up. Jack might be willing to stick around for a little bit longer. But once again, let's thank Jack for an incredible talk.
Thank you very much.